Mattman here, with Standing for Truth. Today, we're going to be giving you one example of why dinosaurs lived recently and are not millions of years old. You see, because of the theory of evolution, scientists have grown up indoctrinated into believing it's the truth without any alternative. This mindset is biased, but none of us really realize it because we see no alternative in school. Unfortunately, it is the paradigm driving the conclusion. They never question the evidence that's contrary to the story because they only know of one story. Rather, they try to find evidence for how the new discovery is somehow wrong and make it fit into the evolutionary story that we were all told to believe without question. One such example is that of amino acids. When it comes to undeniable evidence, we now have something known as racemization rates. If this is new to you, all that it means is that chemical substances come in both left-handed and right-handed forms. Life uses 100% left-handed amino acids to build proteins, and purely right-handed amino acids to build DNA. When scientists in the laboratory toss these chemicals together, they eventually become a 50-50 mixture of both left-handed and right-handed amino acids of that type. When they become this way, all that this really means is that they are now identical in shape, form, and function. They are now mirror images of one another, in the same way as your left hand is similar to your right hand. Now those two forms can change into one another with equal likelihood, so that even if someone began with a pure collection of left-handed forms, in time it will become equally 50-50 mixed, and stay that way indefinitely. Therefore, we can use this observable process as a sort of clock to see how long it has been since a particular specimen has died. This has been done with accuracy in whales. So racemization in regards to dinosaurs is related to the left-handed amino acids that we find in them. You see, over time, their left-handed amino acid profile should have completely converted to an equally left-handed and right-handed mixture. Long ago, if deep time evolution were true, the law of chemistry demonstrates that after death, amino acids racemize back to a 50-50 mixture without question. And the Royal English Society published a time range for when this physical process of racemization takes place. They discovered in most every environment on Earth that in just 100,000 years minimum to 1 million years maximum, all left-handed amino acids will have racemized back to 50-50, even if they were all left-handed to begin with. What do we see when we look around the fossil record? You guessed it. Left-handed amino acid profiles. Exactly what we would expect if young Earth creation were true, and a 100% falsification if deep time evolution were true and dinosaurs really lived over 65 million years ago, using observable evidence. So what do we know? If the vast dates assigned by evolutionists to the various geological layers are correct, any amino acid sample found in these rock layers dated over 1 million years should be perfectly racemized. Yet, this is not what we observe anywhere. Nothing is 50-50. The fact that we find these amino acids at all in supposed rock layers of all ages is evidence that they are not as old as they claim they are. But the fact that we do not only find them, but are still predominantly left-handed is the smoking gun. One perfect example is the chert layer, known as the fig tree chert in South Africa. It is estimated to be 3 billion years old, yet it contains only amino acids in their left-handed form. In the United States, in Green River, Wyoming, they found oil shale, which was estimated to be 60 million years old by evolutionists. When they tested these amino acids, yet again, they found that they have not completely racemized either. The evolutionists had no choice but to argue that somehow most of the geological layers have become contaminated with amino acids produced in a much more recent time. We, the creationists, can simply accept the evidence at face value. They, however, yet again, have to invent another pathetic rescue device that combats the observable empirical data. If this is not bad enough, insects preserved in amber also yield mostly left-handed amino acid profiles, such that the researchers pathetically suggested that a half-life of 1 billion years for racemization must be taking place in amber. Talk about a weak rescue device. Without any testing, without any observation, or without any evidence, they can just assert that it must be this way because evolution is true. Again, we see the paradigm driving the conclusion. What do we actually have as far as evidence is concerned regarding preserved specimens entombed in amber? Well, they took what they believed were 40-million-year-old fruit flies and honeybees and compared their amino acid profile to modern-day flies and bees. And guess what? They were not the same. This falsifies their 1 billion-year half-life hypothesis because you cannot claim that the amino acid profile is preserved intact for over 65 million years when we now have evidence that these cannot even preserve them over a 30,000-year period. They have now lowered their amber preservation potential to just 10,000 years as a half-life because of it. Falsification after falsification. So in conclusion, we have observable tests and evidence which show mostly left-handed amino acids in amber, shale, oil, dinosaurs like Tyrannosaurus and the T-Rex eggshells, and every single layer of the geologic column and chert itself. This is just one piece of evidence in the long line of evidences that we will be exposing regarding dinosaurs and how they are not millions of years old. Stay tuned for more, patrons. Till next time.
Everybody, God bless. SFT is out.
And we are back. You didn't think we were going to leave you for that long, did you? This next topic is specifically that of the amino acid methionine. Now remember, amino acids are the building blocks that are used to make proteins. The specific amino acid methionine is abundant in nature since it is an essential amino acid, meaning it was in dinosaurs. What makes this amino acid unique is that it is particularly susceptible to strong oxidizing agents like hydroxyl, and since carboxylic acids are organic acids and abundant in nature, this would quickly oxidize methionine into methionine sulfoxide. Yet, this is not what we see when we investigate dinosaur tissue. For example, the Nature Communications study results in 2015 revealed unoxidized methionine in many of their Alberta dinosaur specimens. So the very first time we actually look for methionine in dinosaurs, we find it. How could this even remotely be possible unless the dinosaur specimens were young? The answer to that question is obvious. They are young, and evolution is alive. Welcome to our homeworld, everyone. Hold your breath, because we are going deep tonight. So far, if all of that evidence isn't bad enough for evolution, DNA, collagen, and cartilage have also been discovered in many dinosaurs as well. Now here's the problem for evolution regarding these substances. You see, cartilage has a very short half-life. The half-life for cartilage collagen was calculated to be only 117 years, and that of skin collagen is just 15 years. That means that after just 10 half-lives, we end up with only 0.1% left. So starting with a living creature with pure skin collagen, we go from 100% down to 0.01% in a mere 150 years. Now, if we are going by cartilage collagen with a half-life of 117 years, then after 100 half-lives, any evidence of it would be unreadable. This is just after 11,700 years. Triple that number, I don't care. Even that would only give you 35,100 years max, and there would be nothing left. This is nowhere near the date that they require for their theory of evolution. But you have to remember, evidence against evolution is met with condemnation and resistance. Even Mary Schweitzer herself was in fear for her reputation and job because of her discovery of dinosaur soft tissue. Just listen for yourself. She admits this.
This was a combination of my absolute worst nightmare and Christmas. Every day in the lab for about a month. I couldn't wait to get to work, but I was scared to death at what had happened overnight.
This was impossible. This bone was 68 million years old.
So you see this and you think, what?
I didn't want to tell anybody. I didn't want to believe it. I was scared to death.
Schweitzer couldn't believe her eyes.
I was scared to death. This is not supposed to be possible. So we repeated this about 100 times and I didn't tell anybody for over a year. I was scared to death.
When investigating bone, Mary Schweitzer and 3 others discovered what clearly looks to be dinosaur DNA in Tyrannosaurus rex and Hadrosaur dinosaurs. Also, 4 dinosaur proteins were identified, including a cytoskeleton component actin and a microtubular building block called tubulin. A paper by Royal Society Publishing hypothesized a half-life for DNA of only 521 years, even in an optimal environment, suggesting that DNA should be degraded to just single base pairs in less than 7 million years. Their half-life estimate in the study was based on extrapolations of data taken from 150 relatively recent bones. They wanted good, pure samples. They confirmed that there is no possible way DNA could last that long, as they were claiming, even through preservation. And they even state, this confirms the widely held suspicion that claims of DNA from dinosaurs and ancient insects trapped in their amber are incorrect. What does that mean? It means that what they're looking at either can't be dinosaur DNA, or it is and it can't be old. That's it. There is no alternative. But if all this wasn't bad enough for evolutionism, the news gets even worse when they tested what the Egyptians used as paper. They discovered the half-life of papyrus DNA is only 24 years. This discovery, outlined in the scientific paper, destroys the commonly held evolutionary argument where they claim that the presence of DNA must be contamination. As far as the data goes, this paper is the smoking gun, falsifying the dying claims that all of these scientists have been confusing contamination for endogenous dinosaur soft tissue. You can hear Mary resort to the age of the fossils strictly by the rocks they are found in, nothing else. The fact that any of this material exists was astounding enough for evolutionists. So, we have mainstream scientific community attacking her findings that she must now defend. So she, of course, uses the made-up geological record stacked with the evolutionary storyboard and says right away, before any of this preservation method stuff might be known, that there is no possible way dinosaurs could have lived recently since they died 65 million years ago. Again, the paradigm driving a conclusion. Of course, this is what we would expect from a dogmatic evolutionary dominant indoctrinated system. She knew her job was on the line, and she knows what happens to people that do say dinosaurs are young. So she only had a few options. One, she could say that dinosaurs were young, just like the evidence tells us, but then her job would be at a loss. Two, she could claim that contamination occurred, which many were saying was probably the case. Three, she could have said that she was looking at biofilms and nothing from the original dinosaur itself.
There's the matrix, parallel fibered, very white, doesn't look very altered at all. There's the blood vessels with stuff inside of them. And what was even cooler about this vessel, those osteocytes with the little legs are still hooked into that white fibrous matrix and still hooked to one another, which is hallmark for human bone cells and everything else because they have to talk to one another. So given this incredibly good preservation, still super controversial, we thought, well, what else can we tell with it? So we attempted to do mass spec sequencing on this new dinosaur, and we were able to demonstrate several peptides that we know are derived from collagen. And that's really important because I cannot shed collagen. It's all internal. I can shed keratin, but not collagen. And microbes don't produce collagen, so it didn't come from bacteria. It had to have come from the dinosaur.
Or four, there is some unknown preservation occurring. Well, after some time in the lab, she ruled out contamination, fungus, and biofilms in her lab experiments. So she was left with two options: follow the logical conclusion and the evidence, which would make her lose her job and credibility, or four, say that there's some unknown process occurring. But she had no evidence for this. So her only option was try to find any method of preservation for any of these materials. I just want you to think about that for 1 second. She chose option 4 without any evidence and without any hesitation. She knew that all the data pointed to the impossible and that dinosaurs were young, but she could not allow the data to speak for itself and say what the evidence was actually clearly showing us. By choosing option 4, she kept her job and her reputation, and all she had to do was just give an answer. Any answer other than dinosaurs lived recently, and the data shows it. Her lucky break finally came. She was listening to a talk on Alzheimer's and how iron acts like formaldehyde. She thought maybe the iron in the blood would also act like a preserving agent. So she took some vials and filled them with iron-rich blood from birds, and then dropped some soft tissue in them. Lo and behold, she discovered that soaking the material in this blood allowed it to stay preserved. 2 years after the soft material has been soaked in this fresh blood in her lab, it still retains its soft pliability. Real quick, here are some of the many problems with this. 1, not all soft tissue is preserved with the presence of iron particles around it. And Mary's experiment is nothing like what we see in nature. Soaking single vessels in iron-rich blood in a laboratory cupboard is a far cry from a bone laying in the ground in mud and dirt for millions of years. Blood corrodes away. Other creatures consume animals, bacteria as well. Or it can even be washed away by water and rain. Now toss that material out the window onto your lawn, and now let's see what happens. Hell, even bury the contents of that vial in the dirt and watch how your experiment fails. She knows that this would fail. It would be a dead end if she tried, but remember, she needs something as a rescue device for her deep time and to keep her job, or else she would end up like Mark Armitage, who was fired for saying that this evidence proves dinosaurs are young. This is the reality. That is why all they have as a rescue device is a pathetic, coddled lab experiment under highly protective conditions to save the soft tissue from degradation, the exact opposite of what nature has to offer. And you will notice that after she came out with this iron preservation hypothesis, the evolutionary community went silent. They didn't fight against it at all. They were actually glad that they had something, no matter how far-fetched of a rescue device it would be. That is why even today, no one challenges her on this. They still focus on trying to prove that she is not actually looking at dinosaur material at all. This is how desperate they are when it comes to this data. They all know that this biological material cannot last this long. They know that iron might preserve some tissue if the right circumstance exists, but not all of it, and definitely not DNA. Matter of fact, iron corrodes away other biological material. But here's the thing, they also know that the data is actually a lie. Because what she doesn't tell the public about testing iron on preserved material is that Mary destroyed the red blood cells to release their trapped iron, then bathed the modern-day bird bone tissues with the purified and concentrated iron-rich matrix. But the Nature's Communications results clearly showed blood cell shapes still intact. So this is just another example of Mary's desperation in trying to answer the massive problems that they face in regards to dinosaur soft tissue. All of the other scientists know that eventually this data will surface and be exposed, and they just want to say that the material was never dinosaur to begin with.
In science is really, really geologically slow to change paradigms, and that's kind of what we're trying to do. It's expensive. We don't have any money. It's time consuming, and it is really controversial. You have to have a really thick skin.
You mentioned that a scientist needs to be thick-skinned,
Uh-huh.
and I kind of hinted at it, but I wondered if you could elaborate on that.
Paleontology has used the same methods for the last 300 years, and paleontology rests on the assumption that there are certain things we're never going to know about dinosaurs. So they don't go out and learn the methods. They don't know how they work. They don't know how to critique the methods. They don't know how to do the controls. They don't understand the science that underpins what we do. Those are all biological questions. Cell biologists have been using them for years. Don't really do the work to figure it out. They're not willing to try it. I was just at a conference in Denmark not too long ago, and it was in front of a cadre of scientists that hate what I do and have trashed me in the press. And so I asked him at the end of my presentation, I put up all the data that we had so far that I could talk about without killing them, and I said, can you tell me what's wrong with my data? They said, well, we just don't like antibodies. We think they're nonspecific. And I said, well, okay, have you ever tried any of these methods? Well, no, because they're not going to work. And I said, okay, have you ever tried taking one of your archaeological bones and dissolving it to see if there's soft tissues inside? Well, no. But we know your stuff doesn't work.
I want you to look for yourself at the vast array of different preserved specimens that they have found.
So this is an example of a 35 million year old beetle that is preserved with color intact. Nothing's happened to it. And how about this guy? Would you expect that guy to preserve in sand? There's no hard parts. It's a spider. I don't like spiders, but I think it's pretty cool that they can preserve. This is a weird one. Anybody notice anything really weird about this guy? Unexpected for a 30 million year old fossil? Yes, organs are present. Skin is present. The femoral arteries are there. What's missing? No bones. Now, how many of you would think about that and predict that from the fossil record? This is some microscopic sections of some of these materials that came out of this Miocene deposit. And if you look really closely, you can see that the bone microstructure is preserved pristinely, and there are sarcomeres present in that banded muscle tissue. Incredible preservation. Feathers. We know feathers preserve in the rock record. How about this? That's embryonic skin from a sauropod dinosaur from Argentina that is still encased in the eggshell. Embryonic skin is incredibly thin, even for a dinosaur, and normally would rot away in about, 3 to 6 days. And this is one of my favorites. These are plants that come from Canada. You can hold them by the stem, and looking at them under the microscope, under the SEM, you can tell whether the stomata on the leaves are open or closed. So you can tell when they fell off the tree. Preservation is pretty amazing. And a lot of the rest of the stuff that we thought we knew about fossilization is really messed up when you look at things like this. That is a trilobite, preserved in 3 dimensions. And it has a friend. And we cannot really explain how these things can persist for probably 400 million years, preserved like this. Here's another one. How many of you knew that behavior preserves in the rock record? We've got this oviraptor, and it is brooding its eggs, protecting its eggs by spreading its arms over the surface. It's missing its head. Probably did not come by that really happily. This is embryonic bones inside the eggshell, and if you look at the top there, you can see a little tiny embryonic skull with little tiny embryonic teeth, preserved pristinely. Here's our pregnant dinosaur. And again, we can tell fibrous matrix. It's flexible and stretchy, transparent blood vessels, and there's a lot of them, and cells with little filopodia.
Now, after looking at all those massively diverse creatures, some of which don't even have blood, can you logically tell me now that iron is what's preserving all these different tissues and cells and organs? Clearly, they are so well preserved that they have not been buried for millions of years. The simple logical conclusion is the truth. Now listen to Mary herself. She admits that these things are still changing right in front of her very eyes.
We have the fibrous matrix. We have what sure looks like vessels. We have what looks like cells. And we begin to notice some different things. So we've got the transparent, flexible, branching blood vessels that we saw all along. But we also have these crunchy blood vessels that are infused with iron and very, very brittle. So they're changing.
Clearly, you cannot claim that something has been preserved over millions of years when they are still changing right in front of your eyes.
It started back in 2000 with a series of coincidences. A member of Jack's team, Bob Harmon, wandered away from a dig site one day to eat lunch and noticed a small piece of bone sticking out of the side of a 50-foot cliff.
I could tell pretty much what it was from where I was sitting, that it was a T-Rex metatarsal.
How was it sticking out? You mean it was the side, here's a cliff, and it was like a little jutting out?
Yep, exactly.
He got a folding chair and he stacked it on these rocks right there. And you can see that this is on the sheer side hill of a cliff.
This is not possible.
No,
is he attached to anything?
No, he's not. That's Bob.
What happened next happened by mistake. Mary put some fragments of the bone in acid to dissolve away the outermost layer of mineral. But the acid worked too fast and all the mineral dissolved away. Being a fossil, there should have been nothing left. But there was, and it was elastic, like living tissue.
This is the piece.
No.
She showed us video she took under the microscope.
That's really what happened?
Yes.
That's the dinosaur bone?
Without mineral now. That's what was left.
It looked like the soft tissue she would have expected to find if it had been modern bone. This was impossible. This bone was 68 million years old. In sample after sample, they were there. Things that looked suspiciously like flexible, transparent blood vessels. She finally mustered the courage to tell Jack.
She said she dissolved the bone away and there were blood vessels. And, I was like shocked. I mean,
how could that be?
How could that be? That's right.
The things Mary was finding inside dinosaur bones,
Look at that.
blood vessels, and even what seemed to be intact cells, pose a radical challenge to the existing rules of science. That organic material can't possibly survive even 1 million years, let alone 68 million. Mary, Jack, and their team published their B-Rex findings in a series of papers in the journal Science and were promptly attacked. Critics said their samples might have been contaminated, or that the supposed blood vessels were actually something called biofilm, a type of slime. But as Mary showed us, she's been able to replicate her findings. These are pieces of an even older dinosaur, a well-preserved 80-million-year-old duckbill. When she dissolved it away in acid...
Let's put this under the scope here.
Well, look. Is that a blood vessel?
This is a blood vessel.
You're
You
kidding.
see the branches right there? And look at all of them. And it's so consistent over and over and over again. We do this bone and it comes out and I get excited every time. I can't help it.
Mary published her new results last year. The controversy still rages, and the stakes are high. If blood vessels can survive 80 million years, what about DNA? Jack Horner is looking. His crews are now wearing surgical gloves, unheard of in the world of paleontology, where no one used to worry about getting skin cells, sweat, even an occasional spilled beer on fossils.
And I think the data is pretty strong right now with everything we've tried. Scientists that criticize our work really only criticize like one aspect of it and just conveniently ignore all the rest of the data. So I don't know. I guess making them do it themselves.
She and her team ran more chemical tests on that tissue and came up with a hint of Jurassic Park, a trace of possible dinosaur DNA.
We were able to show that there's something inside those cells that's chemically consistent with DNA. It reacts to DNA stains and it reacts to anti-DNA antibodies the same way modern cells do, only greatly reduced in abundance.
Finding the soft tissue opened the door to a new world of possibilities. She now set out to do something that no one had ever done before. To try and find proteins, the building blocks of life. She started with this T-Rex bone cell. If there was a chemical signature of ancient proteins, it should be hidden away inside. Mary took a classic tool of modern biology, one that helps to identify proteins in chicken bones, and she applied this same test to the T-Rex soft tissue. If there were no proteins in the cell, the slide on the right would remain black. Anything green would be a sign of life. The green glow made paleontological history.
It was very exciting. Yes, I was very happy. Very cool.
Have you found any DNA evidence in these materials?
It depends on what you mean by evidence.
Well, have you found genes or pieces of genes?
I don't work with DNA. I kind of think DNA is a little boring. I know that's like heretic, but sequence we have not. But again, I haven't really tried. I did send some of this stuff off to my colleague who works on ancient DNA. I don't have a lab for it, and but we isolated those osteocytes. And in the process of doing a lot of other experiments, we found out that these cells are altered somewhat, so they're infused with iron. And if we remove the iron from those cell membranes, we get staining. So we did propidium iodide, which is an intercalating stain. It mixes with the fingers of DNA. We did DAPI, which is another stain that only works with double-stranded DNA. Propidium iodide only works with dead cells, which we would hope those are. And then we used anti-DNA backbone antibodies, and we used anti-histone antibodies. So 4 lines of evidence, they all show exactly the same patterns in the cells. And I didn't put that in this talk, but I could show it to you. And but it's not sequence data, so it's not consistent with anything else, the way that the staining pattern is arranged. So I can say the data support the presence of DNA or a chemical like DNA inside those cells. But the only thing that's going to convince anybody is dinosaur DNA sequence, and I'm not being part of that show. No thanks. And I said, okay, have you ever tried taking one of your archaeological bones and dissolving it to see if there's soft tissues inside? Well, no. But we know your stuff doesn't work.
Now listen to microscopist Mark Armitage explain how a vast array of different soft tissue is now being found and how iron is not even in the location of these different tissues. So how could it preserve them?
This fossil came from Glendive, Montana. This was our second dig, and this is a Triceratops condyle. Now, condyle is a bone that is at the very end of the vertebral column before the skull. Remember, a Triceratops column is horizontal, right? So the condyle is this big softball-sized bone that hangs off the end of the vertebral column, and it supports the weight of the largest terrestrial skull known, the Triceratops. So this is an important bone. It's under a lot of tension and stress. It supports the whole cranium. So we were very fortunate to collect this, and we decalcified it, which means we put it in a weak acid. The bones act just like normal bones. They dissolve in this weak acid, just like your bone. If you gave a biopsy at the hospital, they would do the same protocol. Soak it in this acid, look at the soft tissues after the bone mineral falls away. And so that's what we did with this bone. We have found nerves in Triceratops condyle. And so we published this last month in March, and the diagnostic feature is clear. Now it's actually even clearer here. Why is that? Well, because this is lipids or fats, there's a term in forensics called grave wax. These things concrete on themselves over time. They kind of collapse and solidify. And that's one of the reasons why it's kind of hard sometimes to see the structure because of the deformity. But in this case, that cross-weave pattern was pulled apart a little bit as it desiccated and dried. And so we're actually seeing the connective tissue layer, this crosshatch pattern, very clearly. Not only did we find that, and we did find many examples of this, we were prompted by this work to then go back and look at over 200 slides that we had previously decalcified from six, I think, different individuals, all Triceratops. We found nerves throughout all the slides that we had previously prepared. So we now feel that this is common, as common as the blood vessels are. Not only did we find the nerves, but we were stunned to find the actual sheath separate floating in the decalcification solution. So this is now the sheath of connective tissue without the fascicles in it. And so we can really study the rotation, measure all this, study all the structure in it. It looks really cool in polarized light. Remember I showed you the fascicles, how they move down, and this you can clearly see the wrapping in these. And we found many examples of this different wrapping. So we know we're looking at dinosaur wrapping, nerve wrapping. We know we're looking at dinosaur nerves. And we found fascicles, lots and lots of fascicles. This is a chicken compact bone fascicle. So remember those wavy fascicles that were moving? This is one of them. It's at high magnification. And you can see what in here, class? Bands of Fontana. Yeah? See them? What do you see in the condyle fascicle? Bands of Fontana. Same in the vertebra. This is a vertebra from Triceratops. We have found fascicles in every bone that we have examined for fascicles. And that's like 10 bones, 8 bones so far. So we believe that this is showing us that there's a lot of work to do. And so we're very excited about it. The last thing that we did in this paper is we took that nerve and we thin sectioned it as I mentioned it to you. Like we did with the chicken nerve, we thin sectioned it. But we made what are called serial sections. So this is one section right after the other, right after the other. It's hard to do, let me tell you, because these sections are 1/100th the diameter of a human hair, and a breeze will carry it away. So it's really careful work. But look at what we showed here. You can see the outer connective tissue layers with the grooves in them. Here's the grooves here, and they're showing up in the thin section now. We're seeing a fascicle underneath the connective tissue with the bands of Fontana in it. And the final section shows one vertical band of Fontana left from the fascicle that was sectioned. So we know we found nerves. We know that this is from the condyle. We expect to find it in just about every bone we look at. We were honored in a great, great way for this paper. They gave us the cover of the journal. They recognized the work, and they're honoring it. The last time I was here, we talked about a paper that we had just accepted for publication. We spoke in August, right? And then it was published in September. And in that paper, we published our results of blood clots in dinosaur bones. And so this is a picture of one of those bones. And it has been thin sectioned. So we take a bone like this, we put it in a special machine, and it shaves it down to about 1/3 the thickness of a human hair. So it's a very laborious process, but we get it thin enough so that we can examine it under the microscope. Now, you see the bone is kind of brown, yellowish brown here. And this ages over time. Even 100 years in the ground would cause this kind of bone material to kind of darken a little bit, right? But this here is excessive. See all this black stuff? And it's all in the canals. These are blood vessel canals that go through the bone. So you have canals in the bone, and the blood vessels and the veins and the nerves actually travel together inside those canals. This is all clotted blood, folks. It's all clotted blood. And so what we showed was that the blood, which has iron in it, is still present in the canals. This is a bright field picture, so now we're pushing light through that thin section of bone. So this is all light coming from the microscope, and you see how light it is, but this is all black. This is a blood clot, and look at how much of the total volume of the vessel canal is cut off. This is rare, actually, to see this much of an opening in some of these clots. Most of the clots that we find are fully filling that lumen, that opening of the canal. This is what it looks like under a regular bright field microscope. But then we used a special microscope that works with UV light. It's very dangerous for your eyes, so we have to really protect ourselves. But the advantage of the UV light is it makes the iron autofluoresce. All tissue glows if you hit it with a certain energy, a certain wavelength of light. All tissue glows, okay? And so we take advantage of that property and we hit it with UV light, which makes the iron react, autofluoresce, give off its own light. And it does it at a characteristic wavelength. So we know we have iron, and it's right here in the canals. Now, I'm not going to take a long time to talk about it, but there is a theory that iron preserves the soft tissue. How many have heard of that? The iron preservation theory? A couple of you. Make a peanut butter and jelly sandwich next week, put it in the backyard as an experiment, and measure how long it takes for that soft stuff to go away. And you'll get an idea. We know innately, right? Soft tissue doesn't last. So it kind of begs the question of age, doesn't it? So this is then the work that we published in Microscopy Today in September. We found it in the horn, the rib, and the frill. And in fact, it was 6 individuals that were separated from each other in the digs who had clots, all triceratops. Well, we've done more work of that on that, and I'll show you that in a second. But I've discussed a little bit of the significance of why this is important. There's a medical condition that you may have heard of called disseminated intravascular coagulation. Who would repeat that to me? No takers, right? It's hard. It's a known medical condition in the medical literature that happens when a human drowns. When there's a human drowning victim, when they die through drowning, their blood stays clotted in their soft tissue and in their bones. You get clotted blood when you die from asphyxiation in water. If you survive and you're revived in the ER, which happens a lot, you bleed out. Why? Well, there's this process of coagulation that you've heard about. It's a one-way reaction. It's dependent on the reactants. When clotting begins, it's a one-way reaction. It goes till all the reactants are gone. And if you die, you stay in that condition. If you survive, it starts to go backwards, and instead of clotting, you bleed out. So now these ER physicians and nurses are trying to save your life while you're bleeding out because they resuscitated you as a drowning victim. These guys weren't resuscitated. So their bones are full of clots. This is important because the clots sequester the iron from what are called Fenton reactions. Don't worry, I'm not going to do chemistry on you today. Picture Pac-Man running around eating those pellets. Fenton reactions needs water to create the Pac-Man that go and eat the tissue. And boy, does it eat the tissue. How many of you had a steel bumper in Detroit during the winter in the past? You know what I'm talking about. Iron eats the steel. Imagine what it does to soft tissue, and it is highly destructive. So we're showing that the iron is left in the canals. It didn't migrate out of the canal into the bone, right? You can see that, can't you? So the iron was unavailable, and if these vessel canals are clotted up and blocked, the water is not available. Water is required for Fenton reactions. So we think we have a great answer to this iron experiment that I wish I had the time really to talk about the experiment, because it is really not the kind of experiment you'd want to conduct to prove that iron is the answer. Everybody's acting like iron is the answer, but they haven't proven it. In fact, the structures that I am going to show you today, that nerve that you just saw, iron doesn't affect it. So it's hard to explain this iron experiment based on the findings that we're making. So we think we're making headway scientifically, and we think that's important. Why do I mention all that? Because we decided to process some Nanotyrannus bones that we collected in Jordan, Montana, May of 2016. We collected a rib, and we collected a vertebra, rib and a vertebra, and we thin sectioned those like I just showed you. And you can already see some of the black in there, can't you? So we found clots in Nanotyrannus. Again, this is a bright field picture, but I'm using both the UV and the bright field at the same time. So you see the light coming through the bone, and then you see the iron reacting. If you turn off the bottom light, it looks like this, and I kind of made it monochromatic so it's more easy to see. But all of this iron in here is returning a signal saying, I'm here and not here. See that? So Nanotyrannus is behaving the same way as all those Triceratops specimens did. Here's another shot of that. There it is, monochromatic. Look at all this beautiful iron just pooled in here. And it didn't cross the barrier into the bone. It didn't migrate into the bone. So what preserved the tissues? If it wasn't the iron.
Like, I see the pictures and I go, well, yeah, sure, there they are. But you're saying that's not enough.
We haven't produced it yet. I have no idea what it's going to take to convince them. Because if it's not sequence data, morphology, histochemical staining, and antibody binding, and all the other methods we've used, I'm kind of running out of tricks.
When it was first published in 2005, this research wasn't universally accepted. Some scientists said her samples might be contaminated. Others were dismissive.
Yeah, I had a reviewer on one of my papers once say that he didn't care what the data said, he knew it wasn't possible. And for me, it's like, if you can't be convinced by data, then how is this science?
Notice how Mary says, how is this science? Exactly! Evolution is not science. It doesn't allow evidence that falsifies it. That is why Mary had to find a solution for how something can preserve this soft tissue, no matter how absurd, no matter what it was. Now, let's switch gears real quick. Is there any other irrefutable evidence that man lived with dinosaurs? Like, for example, has a human ever been found alongside of a dinosaur in the fossil record? Yes. As a matter of fact, this has been found. Most people have never heard of the Ashley phosphate beds of South Carolina, which is a shame because it is the best piece of evidence to ever exist currently that man has lived alongside of dinosaurs without question. The Ashley phosphate bed is the largest graveyard ever found. This information has been forgotten by the sands of time, but I am going to bring it back. Today, the Ashley phosphate bed is mined for its high quantities of phosphate, which is being used for fertilizer, so there is great money to be made. This sedimentary layer is huge, covering large portions of the Carolinas and extending into Florida all the way up into Canada. World-famous geologist at the time, Louis Agassiz, described the phosphate beds as the greatest graveyard he had ever known. The phosphate beds are so full of bones that it is believed by scientists that the phosphate itself is made from bone. So, if the Ashley phosphate bed is loaded with such an incredible amount of fossils as to cause the leading geologist and zoologist of his day to call them the greatest graveyard he had ever known, you should ask yourself, why has zero mention of this ever been brought up? The fossils completely ravaged the evolutionary timeline. That's why. Remember, if the geologic column is out of place, even in one area on Earth, then it falsifies the theory of evolution, and that the layers form slowly and that they represent time. Well, when you look at this one place where there's an 18-inch thick layer, you will see why it's suppressed. In it are rare fossils of everything from whales and porpoises, fish, sharks, megalodon shark, toads, crocodiles, alligators, turtles, rabbits, monkeys, horses, camels, elephants, mammoth, mastodon, rhinoceroses, giant sloth, muskrats, deer, pig, dogs, hadrosaur dinosaurs, iguanodon dinosaurs, plesiosauruses, ichthyosaurs, and human bones, teeth, and artifacts, all within the same layer. Professor S.F. Holmes, the paleontologist and curator of the College of Charleston's National History Museum, described the fossil graveyard in a report to the Academy of Natural Sciences. He stated, "Remains of the hog, the horse, and other animals of recent date, together with human bones mingled in with the bones of mastodon and extinct giant lizards." And this is on record. The occurrence of a hadrosaur dinosaur remains in the Ashley phosphate beds has not been dealt with in the secular science literature really at all. Only 2 sources exist. They have kept it silent, which is typical. The sad part is, when they first uncovered the relics of the human bone, they immediately became afraid. They, without hesitation, considered their jobs to be at a loss, so they said that the bone was out of place to one another. They immediately threw it in the river to be erased from history and hidden from all view. They knew that such a find would cause them to either probably lose their jobs or their grant funding. And this is all the way back in the late 1800s. They were still in fear of the government from evolutionary dogma, as Darwin's theory had now taken hold. The scientists who studied this knew exactly what they had found. This is why their deep concern for finding such anomalies. Professor of Geology and Paleontology F.S. Holmes believed in the conventional evolutionary sequence. So he knew full well that humans and dinosaurs should not be found in the same layer. In fact, he wrote, "Not long after finding the above-named relics of human workmanship and engaged in our usual visits to the Ashley bed, a bone was found projecting from the bluff. Immediately in contact with the surface of the stormy stratum, we pulled it out and behold, a human bone. Without hesitation, it was condemned an accidental occupant of quarters to which had no right, geologically, and so we threw it into the river. Alas, we have lived to regret our temerity and rashness. A year later, a lower jawbone with teeth was taken from the same bed, and now we have it in the cabinet." After the news became public, I guess Holmes saw no reasons to cower from his discovery anymore, as Holmes portrayed a dinosaur with a man on the cover of his own book. Evolutionists have admitted that if dinosaurs are ever found with humans, then they would admit that the Noah's flood was real and that man did live alongside of dinosaurs, and that the biblical timeline is true. Because this evidence would validate that the Earth is young and that they had been lied to about our ancient history, there is no rescuing device for them either, because the Ashley phosphate beds had no layers to them. All of the fossils are mixed together in one huge pool of bones, 18 inches thick, covering 200 miles, with no layers found anywhere. The experts who spent considerable time excavating in the phosphate beds have said that there are no layers, and not even a remote resemblance of a layer. It is all one graveyard. You can learn more about the Ashley phosphate bed in a book called Man, Dinosaur, and Mammals Together, written by the late geologist John Watson, available at Mount Blanco Fossil Museum. A statement from the book says, since remains of land animals and sea animals were found together, a catastrophic event would have included the burial of enormous numbers of animals, and their subsequent conversion into the rock phosphate over a region of at least 200 miles by 30 miles in extent, which is only partially exposed apart. Such an event is of the order and magnitude of the worldwide Noetic flood. What about visual art with dinosaurs depicted in them? Is this good evidence? I would say yes. This is probably one of the most simple and basic evidences that dinosaurs lived with man. This is the original photo, found in Utah. The Native Americans drew it on a stone. They actually used a pecking mechanism to pound this into the rock. Anybody can look at this image and realize that it's a dinosaur. You could show it to a 5-year-old and they will recognize it as a dinosaur. It even has a face on it. You can see the mouth and eyes. When a skeptic looks at this, however, they don't see it. I don't know why, but they don't. Matter of fact, a lot of them say no, what they were actually drawing was probably just a lizard. Well, I am not convinced of this argument, because look, you can tell they know what a lizard looks like. Here's a goat. Here are some snakes. Yet, look at this. So I believe they drew what they saw, and this video will be my theory. See, to make rock carving images, ancient natives used an object to chip away the original rock in a process called pecking, which leaves behind tiny holes or divots. In a recent report, researchers tried to discredit this interesting depiction of a dinosaur by saying the legs are not part of the image, they are not pecked or otherwise man-made, but they are stained with mud or some other light-colored material or mineral. As their sketch here shows, they maintain that what looks like a dinosaur is really just a snake glyph with a stain below it. Evolutionary biologist Phil Sinter co-authored a paper in an online journal, asserted the theropod-like image was not what it appears to be. They wrote that it is a composite of two separate petroglyphs, and that its apparent legs were created by a mud or mineral stain. They claim that this artifact is now officially discredited, which of course would be the sigh of relief news for evolutionists and dinosaur age, and bad news for the creation model of Earth's history. Fortunately, since the method used in the study was basically done just by looking at the artifact, virtually anyone can cross-check the author's observation, which is exactly what has been done, with common sense testing. But first, they lie about the lower body portion only being stained, and no peck marks making up the lower body, as peck marks extend down into the presumed body of the petroglyph, even extending down into the area where the legs are, contrary to what they tell the public. Here are some pictures of this. Daryl Robbins documented this petroglyph in the summer of 2010. The body and leg area pecking divots were obvious to Robbins, who after reading Sinter's report replied, "From my picture, it is clear that the whole dinosaur shape is chipped into the rock surface, not just a mud stain. It's pretty sad when a lowly IT guy does better scientific investigations than scientists. There is a difference between being debunked and being declared debunked. This petroglyph has not really been discredited as a dinosaur except through mass media. They know that the average person cannot access these easily without a ladder to see them for themselves, so they know that they can just get away with saying what they want and people will just believe it." So upon a huge ledge where this petroglyph is that depicts this large dinosaur, Jan Cameron did an extensive rock art rubbing of the dinosaur image. She was commissioned by a Canadian government to do a rock arts rubbing on fossils because it picked up details that the human eye cannot see. Her scientific analysis absolutely reveals that it is a dinosaur. The dinosaur image was made using a hammer, stone, and a deer antler tint. They would peck hundreds of holes to get the proper texture and create the image seen still today on these rock formations. So when someone can walk up, place a sheet of thick paper over the image and rub chalk over it and get the perfect image of a dinosaur, including using USB portable microscopes and multiple gauge tools to help verify that it is indeed what it looks like, then why believe the so-called experts who just walked up, looked at it, and wielded some story without conducting any scientific experiments? Oh, that's right, never mind, don't question them. They're the paid true experts. Now, let's give them this and say that the image of this dinosaur is not a dinosaur, and it is a complete fraud, even though that's not true. Now we have a huge problem as well for them, because there's a second sauropod dinosaur immediately to its right of the first, with its head turned back, looking at the other, and its tail going underneath the neck of the first. So now they have to discredit two dinosaurs, which they never show nor talk about in pictures, both with peck marks that are made up of their entire bodies, not stains. Stack this with the other evidence of Native American artifacts and the scientific discovery of sauropod coprolites in the area, then the evolutionary dogma of 65 million year old dinosaur crumbles. I urge you to return to common sense. If you want to let them sprinkle on academic stupidity dust on it and just let them lie to you and try to jam another rescuing device down your throat, trying to tell you it's anything but what it obviously is, then be my guest. But the obvious peck marks throughout, the clear visual outline, the rock art cast rubbings made, the fact that there are two dinosaurs side by side, and the many photographs taken by many different people that all show it's clearly a dinosaur by anyone who has common sense looking at these things, then I think it's pretty safe to say we don't need the indoctrinated fear-mongers to come along and try to tell you it's all made up. I fear evolutionary paleontologists are highly indoctrinated, so they cannot see the obvious sometimes right in front of them. They can suffer from paranoia because the current living dinosaur is a paleontologist's nightmare. It is a smashing blow to evolutionism, which means they would have to admit what they have told everyone was wrong, what they teach is wrong, and what they believe themselves is wrong. Their cognitive dissonance will never allow it. I urge you to investigate Native American legends and artifacts depicting dinosaur-like creatures. It should leave your mind far more open to the possibility that humans depicted what they saw, because no other logical conclusion could be made as to why people worldwide made up such things. But it gets better. The Doheny expedition documented the fascinating petroglyph of what they even labeled as a dinosaur. Hubbard commented that the photograph of the dinosaur, petroglyph, had been shown to a scientist of report, who then remarked, "It is not a dinosaur. It is impossible because we know that dinosaurs were extinct 12 million years before man appeared on the earth." Now, one more time, look at that date again. You hear that, everyone? They knew dinosaurs were gone 12 million years before man. Not only is that comment utter proof that their mindset is driving their conclusions, but the laughable, arbitrary 12 million years is comedy gold. Today, that date has changed to 65 million years, and they also claim it's a fact as well, just like they were claiming that 12 million years ago was a fact back then. You can't make this stuff up. Too funny. In conclusion, look at what the Doheny scientific expedition concluded. The fact that some prehistoric man made a pictograph of a dinosaur on the walls of this canyon upsets completely all of our theories regarding the antiquity of man. Facts are stubborn and immutable things. If theories do not square with the facts, then the theories must change. The facts remain. Notice that they agree that even one piece of evidence that contradicts the theory is a falsification of the theory. If only those who believe in evolution only cared about real science and the truth, how different of a reality would we live in today? Evolution would be laughed out of the classroom.
And I hope you all enjoyed this video. Goodbye, everybody. SFT is out.